We're now in the last section of our Fastidious gram-negative bacilli lecture. And in this section, we are going to focus on the genus Legionella, and specifically the species Pneumophila. Now, Legionella pneumophila is the most notorious species of Legionella that is associated with human diseases, and this is the organism that causes legionellosis. There are several other species of Legionella that we will not discuss because they don't cause a significant portion of human disease. Now, the virulence factors is Legionella is one of the organisms that is able to survive and multiply within macrophages. Now, macrophages are phagocytic cells that normally kill engulfed bacteria. However, Legionella and several other organisms, another one we've discussed is Francisella, are able to survive as well as multiply within a macrophage. Legionella also can produce proteolytic enzymes. Now, the organism enters the respiratory tract usually through inhalation. It can also get into the body through wounds. It generally causes a pneumonia, so there's an inflammatory response in the alveoli and bronchioles. It is an intracellular bacteria where it can go in and live inside macrophages, which allows it to be shielded from the host antibody immune response. So antibodies will not be able to go and attack the organism because it's inside the host macrophages. And it can spread from the lung and enter into the bloodstream, causing bacteremia. We're going to talk about the clinical manifestations of Legionella. So, Legionella pneumophila causes Legionnaire's disease, and this is an acute pneumonia. It was recognized in 1976, and how it was recognized is there was an American Legion conference in the late 70s, and numerous individuals at this conference ended up being hospitalized with a pneumonia. Several of these individuals ended up dying. And so when it was looked into epidemiologically, it was found that the majority of people that went to this conference were middle-aged men. And they found that the people that got more severe disease tended to be smokers or alcoholics or had some type of chronic pulmonary disease such as emphysema, chronic bronchitis, asthma, things like that. Legionnaire's disease is a multi-system disease. The symptoms include coughing, chest pain in the chest area, like lung pain, and of course your pneumonia. And it can be fatal in immunocompromised adults, especially the severe infection tends to be more highly associated with men. Legionella pneumophila also causes Pontiac fever, which is a much more mild disease than Legionnaire's disease. It is an acute, self-limiting infection. There's no pneumonia, usually a fever, headache, some dizziness, muscle pain, and it is a non-fatal infection. Other infections that Legionella can cause, it can cause abscesses, postoperative wound infections, hepatic or liver abscesses, pericarditis or inflammation around the heart, myocarditis, bacteremia if it gets into the bloodstream. The organism can be treated with erythromycin, and erythromycin is good because it can get inside of the host cells to kill the bacteria. Now, the natural habitat of Legionella is that it is an aquatic organism. It lives in lakes, rivers, hot springs, moist, mud, soil, but that has to be moist. And it is found in contaminated sources. So it's found in shower heads, it's found in air conditioning cooling towers, as well as misting machines. So when you go into the grocery store and you happen to be standing in front of the vegetable section and you get that misting machine that generates moisture for the vegetables, it has been found there have been some outbreaks that have occurred due to supermarket misting machines. Now, transmission can be droplet aerosols. Commonly, it's usually due to aspiration of contaminated water or a wound that is immersed in contaminated water. So that conference that occurred in the late 70s, the American Legion Conference, and this was in Pennsylvania, what they found was that the organism was transmitted through the air conditioners. So they found the organism growing in the air conditioning water towers, and that was then getting disseminated through the air conditioning system. And these people were sitting in conference rooms, very small conference rooms. It was hot outside, and so the air conditioning was cranking all day long. So the best control for this organism is to disinfect the water supplies, heating the water to 60 degrees, at least 60 degrees, continual disinfection of air conditioning water towers in big buildings such as conference rooms, hotels. And the organism itself is relatively resistant to chlorine, so you have to use other types of disinfectants. To diagnose Legionella in the laboratory, the typical specimen, since it causes a pneumonia-like disease, would be sputum. Also, you can get a bronchoalveolar lavage, a transtracheal aspirate, a lung biopsy, and possibly you can do antigen detection using urine. But I'd say the most common specimen you would get in a clinical laboratory is going to be a sputum specimen. Now, this is a gram-negative, pleomorphic. It has many different forms. There's filamentous forms, coccobacilli to bacilli. It is motile, so it does have flagella, it moves. And the staining characteristics are that it does not gram stain very well. So in order to actually see this organism under a microscope, you need to extend your safranin counter stain for 10 minutes. You can also do a modified gram stain. So you would do your typical crystal violet and your iodine and your decolorizer, but instead of counterstaining with safranin, you would counterstain with a dye called carbol fusion. Now, culture of Legionella is required to definitively diagnose an infection with Legionella. Isolation of this organism is incredibly difficult. It's highly fastidious. It requires ferric iron, a hemin, a cysteine. It is inhibited by sodium ions and aromatic compounds. So the selective media that is used to culture Legionella is BCYE, or buffered charcoal yeast extract agar. Again, you should be going into the lecture notes from week 1 and finding your list of media, finding BCYE and studying it. Any media we discuss throughout this course is fair game on a quiz or an exam question. So the charcoal in the BCYE binds to the aromatic amines that are present. Now, on buffered charcoal yeast extract agar, the organism will grow as a grayish white colony that glistens, and it's usually described as having a ground glass appearance. It is an aerobic organism. It does grow better in the presence of CO2. So you'd want to incubate your sputum. Sputums are usually incubated in a CO2 incubator so that you can get these fastidious organisms to grow. It is non-saccharolytic and it has slow growth. It grows, you won't see a colony for 3 to 5 days. Now, you don't have to speciate Legionella. If you isolate Legionella from a sputum specimen from an individual that has pneumonia-like symptoms, you know that this person has Legionellosis and likely Legionnaire's disease, or possibly they could have Pontiac fever, but at any rate, you do not have to speciate it. If the organism only grows on a media that contains cysteine, such as BCYE, and will not grow on your typical 5% sheep blood agar, then that is classic for Legionella. You can use other diagnostic methods. You can use DNA probes, direct immunofluorescence, indirect immunofluorescence, ELISA tests, PCR, as well as urine antigen tests. Here's a gram stain of both intracellular and extracellular Legionella pneumophila. And you can see the organism is very difficult to see. It doesn't stain well, so it's very, very light pink. Very easy organism to miss under a microscope. Here it is again, extracellular and intracellular in a clinical specimen. And here's a gram stain, and you can see it is pleomorphic. There are some more filamentous rods, as well as some tiny coccobacilli. Here it is on buffered charcoal yeast extract agar after 3 days of incubation. So it tends to be a grayish white colony on BCYE. So this slide is showing a direct fluorescent antibody stain of a clinical specimen. So on the left you can see a lower magnification where you can see a lot of white blood cells, and you could see the apple green fluorescing little tiny rods. Now on the right in figure B, that is much more magnified, so you can really see the fluorescing green rod in that picture. Now here's a flow chart for the Legionella organisms. If Legionellosis is suspected, you would want to culture your specimen on your BCYE. You likely would also use blood agar, because classically it would not grow on the blood agar, but it would grow on the BCYE. And usually it's going to take about 3 to 5 days. So this is the end of our fastidious gram-negative rod lecture. If you have any questions on this lecture, you can post them on Blackboard.